So this is our final part, actually part 4 of Chapter 12's lecture on the cell cycle. And we left off with talking about cyclins and cyclin-dependent kinases. So again, these are regulatory proteins involved in the cell cycle and in control of it, particularly in control of getting past the different checkpoints in the cell cycle. So to recap what we were talking about, the cyclin-dependent kinases, the CDKs, are pretty much present in the cytoplasm of the cell in a pretty regular amount, a pretty constant amount. But without the attachment of a cyclin to these proteins, they're in an inactive form. So it's really the cyclin that is going to determine whether the actual activation of this complex is working or not. So that one of those is that MPF, the maturation promoting factor, and that particular complex is a cyclin attached to a CDK or cyclin-dependent kinase that is responsible for triggering the cell's passage from G2 into the M phase of cell division or into mitosis. So you can kind of think of MPF as maturation promoting factor, but you can think of it really as mitosis promoting factor, because without that factor present, the cell's not going to get past a G2 phase into metaphase. So this is a graph showing the relationship between MPF and cyclin concentration and its relationship to the cell cycle. So you can see in order for that M phase to be occurring, we need a high amount of MPF, and that's only going to occur when there's actually been an increase in the cyclin concentration in the cell. So you can see again that fluctuations of the ups and downs of the cyclin concentration, which in turn is reflected in the complex being formed, the MPF complex being formed. So when we have low amounts of cyclin in the cell, we have the cell remaining in interphase, the G1, S, and G2. And then when we have lots of cyclin present, we have the cell moving into mitosis. And that lower diagram is showing again kind of that same idea, where we have the CDK in its inactive form when it's not bound to a cyclin. And you can see as the cyclin accumulates within the cell, then it binds with the CDK and gets more and more of that complex, MPF complex, formed so that the cell can get past that G2 checkpoint. The other, before I go on, the other checkpoint going back here that's important is that M checkpoint. And I just want to talk briefly about that one. In that case, it's not actually a kinase cyclin complex involved there, because what occurs here is that checkpoint is occurring between metaphase and anaphase. So as the cell has its chromosomes lining up in the metaphase portion of the cell, so that metaphase line in the cell, that lineup is occurring, the attachment of the chromosomes or the sister chromatids to the kinetochores has to occur before anaphase can begin. And until that occurs, there is not a signal released to get past that M checkpoints. And the reason for that is so that anaphase is going to actually pull apart the correct sister chromatids and pull them into two separate daughter cells. So it's really a regulatory kind of a quality control checkpoint to make sure that anaphase is going to occur correctly and that each daughter cell is going to get one of the sister chromatids. So there's actually a series of proteins, and it's a different type of protein complex that acts as the signaling molecule, and similar to what we learned in chapter 11 with the signal transduction pathways. But that is not going to occur until all of the sister chromatids are attached to, and their kinetochores are attached to the mitotic spindle correctly. So that's just a really great checkpoint to ensure that each daughter cell is getting the right amount of DNA and the right set of chromosomes. So some other signals that are actually external to the cell, because what we've been talking about is a lot of internal signaling controlling the cell, but there's also external signals like growth factors, which are proteins released by certain cells to stimulate other cells to go through division. So an example of that is this platelet-derived growth factor, the PDGF, which stimulates the division of human fibroblast cells in culture. Another type of external signal that you need to be aware of is a density-dependent inhibition, in which a crowded set of cells stop dividing. So there's a chemical released by these cells that is in relationship to how much contact they have between each other, and the more contact there is, then the more of the chemical signal that is released, which is an external signal saying stop dividing because we don't have any more room. There's also what we call anchorage dependence that most animal cells exhibit, and all that means is that they have to be attached to something, a substratum of some sort, in order for division to occur. And what's important about especially these last two control mechanisms is that they tend to be the ones that go wrong when cancer is occurring. So this is showing you the anchorage dependence here, density dependent inhibition occurring, and then and all of those on the left are the normal, and the one on the right is when there's cancer, and you can see how density dependence is not a factor with cancer cells because they're very densely packed, and also there is not the anchorage dependence occurring because they're not anchored to any sort of stratum at all. They're building up on top of each other. So they've lost attachment to what they should be attached to. So those are things that have gone wrong within the cell. They've lost that inhibition mechanism. And I'm just going to end with this final great photographs of the different phases of mitosis in eukaryotic animal cells. And review the structures again. Remember to review and recognize what photographs of the different phases look like. And that's it for Chapter 12.